All right, so the next few chapters are going to deal with some of the various body systems of animals, and we're going to look first in chapter 40 at how animals manage water and electrolyte balance. So it's very important that animals have the correct osmolarity and concentration of water in their bodies, and they do that in various ways by removing wastes, taking in water, excreting water, and we're going to look at some of the ways in which this happens very briefly in this chapter. So focus here will be on the excretory system. So the excretory system in humans and invertebrates involves the kidney, the ureters, the urinary bladder, and the urethra expelling wastes from that system, right? Nitrogenous wastes, we're going to see. So we're going to look in this chapter at what the excretory system is, what the purpose is, what waste is, what the different types of wastes are. Briefly about how excretion takes place in invertebrate animals, the structure of the vertebrate kidney, and the function of the vertebrate kidney. So in order for chemical reactions to take place, they have to occur in water. So the hydrolysis and dehydration synthesis reactions that drive metabolism need water. So you need water to break bonds, you need water to build up, complex macromolecules from their monomers. You need water to do that. So if you have a balance of water and solutes get disturbed, then obviously chemical reactions are not going to take place in the same capacity. And if it's so extreme, then you may see life end. So water is very important. Inside of the water and inside of living things, we have electrolytes. So electrolytes are just any compound that's going to dissociate into positive and negative ions when it's dissolved in water. So you may have heard of electrolytes being in like Gatorade and sports drinks and things like that. And the electrolytes you find in Gatorade and sports drinks are, sodium, potassium, and these things are going to be really crucial in maintaining membrane physiology of cells, because when cells go to transmit an impulse or maintain an electrical signal, they're going to need these electrolytes to pump in and out of them, right? And sodium potassium pump, we're going to look at that a little bit later on in a later chapter. There's the importance of calcium in various functions in the body, right? Chlorine ions as well. So these electrolytes, the balance of these electrolytes, having the correct amount in the cells is very important. So how do we do that? How do we keep them in balance? It has to do with excretion. So in order to excrete waste, animals have to produce urine. So when animals produce urine to excrete waste, they do so with the expulsion of water. So depending on the type of animal, this is going to look a little bit different. So for a freshwater animal, marine, or a terrestrial animal on land. So electrolytes and water move through organisms, through their cells, from the interstitial fluid to their cells, their tissues, through two processes here, diffusion and osmosis, and we measure those two by looking at osmolarity. Okay, so that's the relative concentration, just another term for that. So diffusion specifically is the movement of any uncharged particles down their concentration gradient. So diffusion involves the movement of any non-water, basically, from an area where there's a high concentration of that ion to an area where there's a low concentration of that material. I shouldn't say ion because we're talking here about uncharged usually. Osmosis, on the other hand, is specifically the movement of water down its concentration gradient. So where you have a high concentration of water, you're going to have a low concentration of some kind of solute. So diffusion is the movement of the solute down its concentration gradient, and osmosis is the movement of water down its concentration gradient. Now, sometimes water can't move. Let's say that the membrane is impermeable to water. Then the solute will move to get to equilibrium. Let's say that the solute can't get through, which is more often the case. Then the water will move to get to equilibrium. Okay? The concentration of the solutes in the solution is the osmolarity, and we measure that in the units osmoles per liter. Okay? So like I said, if the solutes are separated by a membrane that they cannot go through, so that means that it's selectively permeable, that means that the water is going to move from the side of the lower osmolarity to the side of the higher osmolarity. Okay, so water is going to move from an area where you have a high concentration of water or a low solute, a low osmolarity, to an area where you have low water or high osmolarity, high solute. So you can see what this looks like here in a beaker separated by a semi-permeable membrane. So in this case, the membrane is permeable to the solute, those red dots. So over time, this solution is going to reach equilibrium, and the dots on the left side are going to go to the right side until you have a situation where it's all equally diffused on each side. Now, if there's a situation where, let's say, these could not move, so let's say they couldn't pass through, then the water would have to move to get this to an equal, and a whole lot of water here would have to move from the right side to the left side to get this so that the concentrations are equal. So you have to imagine that, this would be very low at the end of that, and then this side would be very, very high. Here we're looking at are three situations where cells are going to take in water through osmosis. So if a cell is in a solution where it's hypertonic, okay, that means the solution has a higher concentration of solute than inside the cell. That means water is going to rapidly leave the cell and it's going to shrivel. In isotonic solution, we have a situation where there's equal solute concentration. So in this case, you're going to see water flow in and out evenly. In a hypotonic solution, you're looking at an area where there's a lower solute outside of the cell than inside. In this case, you're going to see water rapidly in this case, you're going to see water actually going. It should be going from an area where you have a low concentration of solute to an area where there's a high concentration of solute. So these arrows should be going inward in a hypotonic solution. I'm not sure why they're shown there going out, unless I'm missing something. I guess this. Okay, so the arrows there are showing you solute rather than water, which is strange. But in a hypotonic environment, the cell will swell because water is going in. Okay, so those arrows are a little bit strange. I'm not sure why that figure has them like that. It's not usually the way it's represented. Excretion is the process of getting rid of waste. So waste in humans accumulate, in terms of these types of waste, these water wastes. Urine, sweat, and tears are types of waste that can accumulate. There's other types of waste like carbon dioxide that will also end up in these liquids. Nitrogen, removed from amino acids derived from protein. So nitrogen is being an important building block of amino acids as well as from nucleic acids. Amino acids undergo something called deamination, where they break down, and ultimately their waste is excreted in three different forms: ammonia, urea, uric acid, and these end up in urine as well. Okay. So you can see here on this figure the three forms of waste and kind of where they end up and where we see those types of waste in which types of animals. So proteins break down to amino acids, they're building blocks, and nucleic acids break down into their nitrogenous bases and ultimately end up with these amino groups that need to be excreted. So most aquatic animals are going to excrete the amino group in the form of ammonia, so it's converted to ammonia. Mammals, most amphibians, sharks, and some bony fishes will excrete the amino group in the form of urea. Now, urea requires a lot of water to go out with it when it's excreted. Reptiles, birds, insects produce uric acid, which is very highly concentrated, so that's why birds, their urine is that kind of white, thick uric acid. It doesn't have the water with it, so they can better hold on to water, but they produce this really toxic uric acid as a trade-off to that. So it just depends kind of on the species and how it's evolved. So in vertebrates, there's a few other ways in which wastes are removed. So in sponges, they have a contractile vacuole, similar to what we see in plant cells, that contracts to force fluid and waste out. Some protists do not have organelles and actually expel water directly across their semi-permeable membrane, so it just depends on the complexity. Flatworms possess a flame cell system, it's called, two or more branched tubules that terminate with cilia, and the flame cells will expel the waste. And earthworms have a closed circulatory system with nephridia, and the very similar kind of capillary network right alongside their bladder, and they expel their waste through this nephridia pore, which is an opening excretory for excretion on the earthworm. In humans and in mammals, we have various organs involved in excretion. So you've got the kidneys, which connect to the ureters, which connect to the bladder, which is a holding chamber, muscular tube with rugae in it to expand to fill up with bladder, and then ultimately the urethra for the expulsion of urine. So we can look at the structure of the kidney, and you can see kind of, the pathway of urine drainage here. Essentially, when we're talking about this, and I'm not going to get into too much of the structure of kidneys, this is an anatomy course, but, you got the renal artery, and you've got the renal vein, and the renal artery and the renal vein are going to be bringing blood into and out of the kidney for filtration. So the renal artery here is going to bring the blood, pump it through the kidney into these pyramids in there, and inside of the pyramids, we've got tiny microscopic structures called nephrons. And these nephrons are the functional units of the kidney, and they do the job of filtration. And they will essentially take out all the bad stuff that you don't want and only they're going to leave in all the bad stuff and take out all of the good stuff that you do want and ultimately put it back into the body so that it can be sent back out through the renal vein, and the good blood is sent back to the heart to be redistributed, whereas the wastes are going to go through the ureter out to the urinary bladder. So a very cool system here, and we'll take a look at kind of that pathway, like I said. So starting with the arteries, your aorta to the renal artery, and then here we're inside, and we're getting to the arteries of the kidney, segmental, interlobar, arcuate, cortical radiate, afferent arterial, ultimately to the glomerulus, which is this sort of ball of capillaries where the blood is going to very easily pass through into the efferent arterial, and now it's going to get into the nephron at this point, and the nephron is going to filter the blood. And blood is going to continue to move through these peritubular capillaries in the vasorecta, and these are all around the nephron, okay, until it is sent back out through the cortical radiate, arcuate, interlobar, renal, and inferior vena cava back to the heart to be reoxygenated. So in the dotted line there are all the ones that are inside the kidney, essentially associated with the nephron. So here it is. Here's that nephron tubule. And this is a nice kind of figure because it takes you through essentially what's happening. So here's that glomerulus that I was talking about in the glomerular capsule. So this is one nephron here, and the blood is going to leak through this glomerular capsule, and it's going to stay in there. The red blood cells should stay in there, and the filtrate should come out. And through this whole process here, through the proximal convoluted tubule, through the nephron loop of Henle, through the distal convoluted tubule, right? All of the solutes are going to be filtered and reabsorbed until ultimately we get the urine, which is going to come down here through the collecting duct and ultimately make its way to the urethra to be expelled. All right, and that's what this is showing you here, and I'm not going to hold you too much to this. There's a couple of little animation activities you'll do in the mastering that will walk you through what's happening with specifically the nephron and the changes in osmolarity that take place throughout. And it all has to do with the water moving down its concentration gradient and the solutes moving down their concentration gradient. Okay. And that's what this figure is showing as well. Okay. And this figure is taking you through, again, the various osmolarity changes that take place. And you can see that in the descending limb of the loop of Henle, the water is moving out down its concentration gradient. And then here, as we get to the ascending limb the sodium now is moving out, okay, until we end up with here, filtrate that's mostly dilute at this point and is hypoosmotic to the interstitium and is going to be expelled as urine. Okay, so here's the different parts of the nephron and the collecting duct and what they do. So kind of a little application here at the end about antidiuretic hormone, or ADH. So antidiuretic hormone is responsible for the level of reabsorption that takes place here in the distal convoluted tubule. So if you are, it depends on how much water you're drinking. If you have low water intake, if you're dehydrated, or you have over hydration. So antidiuretic hormone, when it is secreted, is going to influence how dilute or how concentrated your urine is. So one of the things about antidiuretic hormone is interesting is that alcohol actually will inhibit antidiuretic hormone, and that's why when you're drinking a lot of alcohol, you have to urinate frequently because your brain is inhibiting that antidiuretic hormone, which, when we increase antidiuretic hormone, you're seeing less water excreted in urine. When you decrease antidiuretic hormone, you're seeing more water excreted in the urine. So if you inhibit that, you end up with this situation even when you're not drinking a lot of water. So that's why you end up feeling really crappy the next day, because you've excreted all that water that you actually needed because you inhibited antidiuretic hormone, which normally would be in this situation here, dehydration, would be holding on to water. Instead, you're dehydrated, but you're still getting rid of water, which is not a good situation to be in, and you can end up with a hangover from that. So you always want to make sure you're drinking a good amount of water if you are know you're going to be drinking a lot. Okay. All right, so that will wrap up this chapter. So like I said, don't worry too much about the intricacies here of the nephron. Just the basics that I discussed will be what you're responsible for. So this slide here, I think, is a good summary, as well as what will be in the mastering. All right. I will see you guys next time.